This is a quick review of Chapter 16, or the lymphatic system. So the lymphatic system is made out of a lot of lymphatic vessels. We start off with lymphatic capillaries, and in Chapter 15 we talked about how interstitial fluid is made. The interstitial fluid is basically fluid that is found in between cells, and it is going to get exposed to a lot of different things, including germs, toxins, and before that interstitial fluid gets back into the blood or into our systemic circulation, it has to go to the lymphatic system to get filtered. We filter the lymph in our lymph nodes and in our spleen, so that interstitial fluid that is in between cells, when it gets into the inside of the lymphatic capillaries, we now call that liquid lymph. After filtration, now that it's bacteria and germ-free and toxin-free, it'll go back into the venous and join the venous circulation. So that's really, what, kind of like in a nutshell, what the lymphatic system is all about. So we have a whole network of blood vessels. We also have lymph nodes, and we have the spleen. And as I mentioned, it's very closely associated with the cardiovascular system. So you'll see that most of the lymphatic capillaries and the lymphatic vessels, they're very closely related to the venous circulation. And then at the end, after all of the filtration is done, it'll dump all of that now clean lymph into the venous circulation. There are actually three different functions of the lymphatic system. So it'll transport the excess fluid away from the interstitial spaces, and like I said, we are now going to call it lymph once it gets into the lymphatic capillaries. The lymphatic capillaries that are found in the digestive system are known as lacteals, and those help in the absorption of fat and fat-soluble vitamins. It also helps in the defending of the body against different diseases, so it is considered a part of the immune system. The lymphatic pathway, we start off with lymphatic capillaries. So the lymphatic capillaries are blind-ended or dead-end capillaries. So unlike the capillaries in the cardiovascular system that has an arterial and a venous end, the lymphatic capillaries don't have that. They just pretty much start off as a dead-end capillary. And you see here that they're closely related to the venous side of the circulation. And then we end up making a lymphatic vessel, which is your afferent one, where the lymph enters the lymph node, it gets filtered, it exits the lymph node through an efferent lymphatic vessel. It'll gather through lymphatic trunks, and then two lymph collecting ducts, one on the right and one on the left side of the body. And then it'll dump all of the lymph into the venous circulation in the subclavian vein. This is just really a diagram because this happens obviously on the right and on the left side of the body. So we'll see how, what the distribution of the lymphatic trunk, collecting ducts, and the lymph nodes as well, how those are distributed within the body. We'll start off with the lymphatic capillaries, and as I said, these are these closed-ended tubes, and you'll find them kind of start with within or from within the interstitial tissue. And you can see here that lymph is going to be will diffuse from the interstitial tissue into the lymph. These capillaries are made of simple squamous epithelium, just like the blood vessel capillaries, and it allows for passage of fluid in one direction only, which is to the from out to in, and it'll deliver all of that lymph to your bigger. It'll kind of gather all these little capillaries into bigger lymphatic afferent vessels. The lacteals, again, in the digestive system, those are the lymphatic capillaries there, and those help in the absorption of fat and fat-soluble vitamins. This right here is a lymphatic vessel, whether an afferent or an efferent. It also has the three layers, just like veins. So there is the endothelial layer, or the inner layer. There's a smooth middle layer, and then there's an outer layer made out of connective tissue. They also have valves just like the veins in our cardiovascular system. And the lymph will be collected, as I said, it'll drain in the lymphatic lymph nodes, and we'll talk about the structure of lymph nodes in a bit, and then exit out of the lymph through the efferent vessels, and then they will gather through lymphatic trunks. Lymphatic trunks, so those lymphatic trunks drain from your lymphatic vessels, and they are scattered throughout the body, and we name them according to the region that they're in. So the lymphatic trunks found, for example, in the lumbar area is known as your lumbar trunk. We have trunks in the intestinal area related to the small and the large intestine. Those are known as your intestinal trunks. There are subcostal, for example, so those are found in the subcostal area, which is, subclavian area, which is underneath the clavicle. There's also lymphatic trunks in the jugular area and so on. So these trunks are widely spread throughout the whole body. And then we are going to collect all of that lymph into two lymphatic collecting ducts. There's one on the right side and one on the left side. The left side is the biggest of the two, and that collects lymph from most of the body. That's known as the thoracic duct. On the right side, the lymphatic duct is much smaller, and it collects lymph only from the right side of the head and neck, the right side of the chest, and the right upper limb. So we can see that better in this image right here, where you have the thoracic duct on the left side, and that will gather lymph from the left side of the head and neck, the left side of the chest, left upper limb, and the whole rest of the body, while the right lymphatic duct collects lymph from the right side of the head and neck, right side of the chest, and the right upper limb. Well, since the lymph nodes in the axillary area drain lymph coming from breast tissue, that is why one of the areas that we have to examine if somebody has breast cancer would be the axillary lymph nodes, because those cancerous cells can potentially get into the lymphatic system and lead to the enlargement of these lymph nodes. After a mastectomy or after a radical mastectomy where these lymph nodes are removed, now that we have lost that drainage of the excess interstitial tissue, that might lead to edema, which would be swelling of the upper limb. So the axillary lymph nodes is responsible for draining lymph from the breast tissue and from the upper limb as well. So if you remove those lymph nodes, that can lead to accumulation of the interstitial tissue in that upper limb, leading to its swelling. So tissue fluid, as I said, that is the excess of it, or whatever we are going to drain, is going to be known as lymph as it enters your lymphatic capillary. And lymph depends on the formation of tissue fluid. So the more tissue fluid we have, the more lymph we have. So tissue fluid formation depends on the different pressures that we talked about in chapter 15. So this right here is that's exact same image. I just copied and pasted it here, where we have that capillary, and right up here is your interstitial tissue. So you have your tissue cells with the spaces in between, that's your interstitial space. And you have arterial blood, oxygenated blood coming in one end of the capillary, and it leaves as deoxygenated or venous blood on the other side of the capillary. So remember we mentioned that on the arterial end, it favors filtration because of the high hydrostatic pressure, where plasma and the smaller molecules are able to filter out into the interstitial area, making interstitial fluid. While on the venous end, because of the higher osmotic pressure, fluid is going to be reabsorbed or sucked into the venous end of the capillary. So whatever is left in your interstitial space makes your interstitial fluid. Now that fluid again circulates in between the cells, and it gathers all of the toxins, the metabolites, any germs that are there, and all of those germs are going to be absorbed or will diffuse into these lymphatic capillaries. So basically your tissue fluid is blood plasma minus your plasma protein. So these plasma proteins are too big. They cannot get filtered out through those little tiny capillary slots. So again, your tissue fluid is basically plasma minus your big plasma proteins. Okay, so we'll talk a little bit about lymphatic tissue and lymphatic organs. So the lymphatic organs are your lymph nodes, thymus, and spleen. We also have these little lymphatic cells or scattered lymphatic tissue known as mucosa-associated lymphoid tissue or MALT. Those are lymphatic cells that are lymphatic tissue that are kind of scattered throughout the body in areas that are lined with mucous membranes. So the digestive system, respiratory system, urinary system, and reproductive tracts. Okay, so example of these are the tonsils and the appendix. Peyer's patches, those are little collections of lymphatic tissue found in the terminal ileum, which is the last part of the small intestine. And so you can imagine that, remember, mucous membranes are the parts of the body that are exposed to the outer environment. So the oral cavity, for example, the digestive system, respiratory system, the nose, and so on. So all of these areas where there is a connection between the outside world and the internal environment of our bodies, we need to have those lymphatic tissue in order to try to capture and prevent germs from entering the body. So take a look at lymph nodes. Lymph nodes are very tiny. They're kind of, this is a real lymph node. This is a depiction or kind of like an image of it. These lymph nodes are very tiny. They're kind of like pea-shaped lymph nodes, and they are going to filter pathogens. You can see these lymphatic vessels that are draining lymph into the lymph node. It gets filtered on the inside, and then it leaves through one of those efferent lymph nodes. Your lymph node has, as you can see here, this is your afferent lymphatic vessel, and this would be your efferent. The area where the lymph exits out and you have the blood vessel, that is known as the hilum of the lymph node. And you'll find that lymph nodes have lymphocytes, and those lymphocytes are going to attack your germs, whether they're viruses or bacteria or parasites. It also has macrophages, which are part of your phagocytic cells. Okay, so that is going to try to engulf or eat up, destroy any foreign bodies, damage cells, and cellular debris. Remember that these lymphocytes or macrophages are not made in the lymph node. Okay, remember those were made in your bone marrow, but then they circulate and kind of end up being living in these lymph nodes. So location of lymph nodes, they are widely scattered throughout the whole body except for the central nervous system. So you're not going to find lymph nodes around the brain or around the spinal cord. But with those, with the exception of the CNS, lymph nodes are found everywhere. And they're like, again, they tend to be in groups. So you'll find, for instance, inguinal lymph nodes. You'll have pelvic lymph nodes, abdominal lymph nodes. There are axillary lymph nodes, thoracic, cervical, supratrochlear, that's related to the elbow, and so on. So they tend to kind of gather around and be in these little collections, and they would drain the more distal areas. So for instance, the inguinal lymph nodes would drain, as you can see, from the whole lower limb, and the lymph would kind of go against gravity. They'll drain, they'll be filtered in these inguinal lymph nodes, and then eventually reach the systemic circulation. We'll see how that happens. First off, we'll talk about the thymus gland. So the thymus gland, and do not confuse it with the thyroid gland that's up here in the neck. The thymus gland is a butterfly-shaped bilobed gland. Also it's found in the chest, right behind the sternum. It's divided into two lobes. It has lots of lymphocytes, and those lymphocytes, again, really originate in the bone marrow. Most of these cells are inactive, but they become activated if they are activated and mature in the thymus, they are going to become T lymphocytes. And that maturation happens through a hormone known as thymosin. So the function really of your thymus gland is to make thymosin hormone, and thymosin hormone helps in the maturation of these immature progenitor cells into T lymphocytes. As we age, though, the thymus gland tends to shrink. So you'll find here that the thymus gland is the biggest. Here, this is the fetal thymus gland, and you see in relationship to the surrounding structures, it's huge. While as we age, as we become adults, the thymus gland tends to shrink. That is because as we're, fetuses and then babies and kids, that's the time when we are exposed for the very first time to a lot of germs, and that's when that gland is most active. As we age, this gland again tends to shrink, and that's why again in senior citizens, they are more exposed, or they are more prone to infections because of the loss of the function of the thymus gland. I've seen cadavers where the thymus gland is actually totally non-existent, and it has been totally replaced with fat cells or adipose tissue and just, connective tissue. The spleen, which is found in the upper left part of the abdomen, we consider it a huge lymph node. Okay, so this is the spleen is the largest organ of the lymphatic system. It resembles the lymph node kind of like in its structure and its function. The only difference is that the spleen filters the whole blood, while lymph node filters lymph. The blood that comes from, especially after reabsorbing things from the small intestine, you do not want that blood you don't want that venous blood to kind of go straight to your heart, to the systemic circulation. You want it to go first to the spleen and to the liver, where they can get filtered and kind of cleaned up of any germs. Okay, so that is going to happen in the spleen. The spleen has a huge amount of blood. It's considered a blood reservoir. So in cases where patients are bleeding profusely, the spleen has the ability to squeeze itself, so to contract those smooth muscle fibers and kind of squeeze the extra blood that's in there. So it's kind of like the patient giving himself or herself an auto transfusion. So inside of the spleen, if you take a look at the microscopic picture of a spleen, there are what are known as two different tissues. We call them pulps. There is white pulp in the middle, and that white pulp is full of lymphocytes or white blood cells, hence the name white pulp. And the red pulp on the outside is full of RBCs, hence the name red pulp. It also has lymphocytes and macrophages, but in order to start kind of filtering those, the blood cells that are coming in. Then we'll talk about the body defenses against infection. So the lymphatic system has different ways, or our body has different ways of protecting us. Let's kind of go over a couple of definitions first. So a pathogen, if you break down that term, patho means pathology, gen means to generate, or to genesis, to create something. So a pathogen is any agent that can create a disease. So that could be a bacteria, a virus, a yeast, a parasite. So anything basically, that can make us sick is known as a pathogen. Okay, we are all exposed to pathogens all the time, but we don't get sick all the time because our immune system keeps the number of pathogens in our body in check. It's not until that number, the pathogen is able to multiply and cross kind of like a threshold level where our immune system has not been able to keep that number in check, that's when we get sick. So we have different ways of fighting off infections. There's an innate or a non-specific defense, and that really does not depend on what kind of germ is coming in. So regardless of the germ coming in, our body will try to defend us using these innate defenses. So it's a very general defense. It does help us, it does protect against lots of pathogens. But we also have a more adaptive defense. So when this innate defense fails, we are going to go on to an adaptive defense, and that is more specific to the germ coming in. It is immunity, so meaning that if I attacked something using my adaptive defense, I am now immune to that germ. And so again, it's more specific, it's more precise, and it's made by the lymphocytes. So we're going to talk first about the innate defenses. The first one on the list is species resistance. Species resistance means that a germ is confined to a certain species and cannot be transmitted to another species. This really has proven to be broken. Okay, so species resistance is still there for the most part, but we have heard of avian flu, like flu that comes from birds. We've heard of swine flu that came from pigs, and so on. So germs that used to be kind of contained into one species and no transmission to another, that barrier is starting to break. We do have mechanical barriers. So just the fact that our skin and mucous membrane are intact, that will form a mechanical barrier that is going to prevent germs from entering into our bodies. It is considered your first line of defense. So remember when we talked about the integumentary system, we kind of mentioned that. And anything else that on these innate or these non-specific defenses are considered second lines of defense. So for example, first lines of defense are the epidermis sloughing off. As we remember, we talked in the integumentary system, the uppermost layer of the epidermis those are dead keratinized cells, and those slough off as we kind of move around. Those are, as they fall off, they are going to remove superficial bacteria that's on our skin. Okay, we also have the ciliated epithelium, the respiratory tract. Those also tend to kind of brush off and sweep away pathogens trying to get into our lungs. We have hair that traps pathogen, like hair in the nasal cavity, for example. And then tears, saliva, and urine. All of these watery fluids help in washing away microorganisms, and all of those are examples of your mechanical barriers. Next on our list of innate defenses are chemical barriers. So we have different chemicals that our body makes normally, and that try to break down germs as they're entering. So we've got some enzymes. Okay, so for example, pepsin in gastric juice, lysozyme is found in tears. So all of these are different are enzymes that are going to destroy these microorganisms. So remember that microorganisms are proteins, so these are really going to digest those microorganisms. Interferon, we will find that interferon is more specific for viral infections. It'll block viral replication, meaning it'll prevent viruses from multiplying. It also protects against the growth of tumors. So interferon, whenever you hear that word, you kind of want to associate it with viral infections and protecting us against cancers. There's a lot of research nowadays using interferon as a way to control and maybe cure cancers. Defensins are from the name, okay, that it's a protein that defends us, okay, it's a neurotransmitter, sorry, it's a chemical that is made by neutrophils, and it'll defend us from these microbes. It really kind of paralyzes them, okay, and kills them by making little openings in the cell membrane. So when you make that opening in the cell membrane, you pretty much killed that germ. Collectins, those are proteins that, from the name, tend to gather bacteria together and make it easier for the immune system to recognize it. So instead of having, 100 cells of bacteria scattered throughout the body, this collectin will kind of collect them together and make it easier for the immune system to attack them. The complement system acts whether in the innate defenses, and we'll talk about them again when we get to the specific defenses. That's a huge group of different proteins, and it'll stimulate inflammation, it'll attract phagocytes, and enhance phagocytosis. It's really kind of like immune system on steroids. So the complement system is a very strong component of the immune system. Other innate defenses are natural killers. So these are it's a very small part of the lymphocytes, and from the name you can see that they're very aggressive cells. They defend us by making perforations. They will release these little chemicals known as perforins, and those perforins are going to make holes or perforations in the cell membrane, and again, that'll destroy the pathogen. Another one of the innate defenses is inflammation. So the signs of inflammation are redness, swelling, warmth, and pain, or RSWP instead of the RSVP. That was our mnemonic. Okay, the whole idea of inflammation is to inhibit the spread of the infection. So it's kind of like to surround the area of infection where the germ is to contain it into one area of the body and prevent it from spreading otherwise. Most of the signs and symptoms are due to the vasodilation that occurs to the blood vessels. So that vasodilation will open up those capillary slits and lead to increasing capillary permeability. So you'll find that the area is swollen. There is redness, warmth, and it's painful and tender. There's a difference between tenderness and pain. Pain means that it's just painful without touching it. Tenderness means that you'll feel the pain upon touching of the area. Then you'll have white blood cells that come into the area. White blood cells are going to start attacking the germs, and then pus might form. Pus is dead white blood cells, dead bacterial cells, cellular debris that has been accumulating because of the tissue damage. That is what pus is made out of. Okay. Then you'll have clotting factors that come in. Those clotting factors are going to, again, deposit fibrin in order to contain the infection in that one area. Then you have fibroblasts that come in. Fibroblasts start the healing process. And then the phagocytic cells are going to engulf any tissue debris, any dead cells, and remove all of that, and then we could heal, complete our healing by cell division. Another one of the nonspecific defenses we have are phagocytosis. So we already know the whole process of that, and the phagocytic cells are your M&Ms. Those are your macrophages, neutrophils, and monocytes. Okay, remember that monocytes and macrophages are basically the same cell. It just depends on where that cell is. So monocytes are the name of the cells when they're inside of the blood vessels. When they leave the blood vessels, they become more active, more hyper, and we are going to call them macrophages. Okay. And the way that the body knows that we need these phagocytic cells to come in are by chemicals that are released by the damaged tissue. That process of calling in the phagocytic cells is known as chemotaxis. Another innate or nonspecific defense is fever. So just the fact that our body temperature goes up, that makes our temperature, it inhibits bacterial growth. So in order for bacteria to grow, it needs your normal body temperature. A fever makes your body environment inhabitable to bacteria. So it will inhibit bacterial growth. Another thing that the liver does when the body temperature goes up is that it hoards all of the iron. It keeps the iron, and iron is something, again, that the bacteria needs. Bacteria and yeast both need in order to grow and make us sick. Also, the high body temperature increases your phagocytic activity, so it stimulates the immune system. Our fever goes up because of a cytokine or a chemical known as interleukin-1. Interleukin is a chemical that acts in between your white cells, so it's communication between white blood cells, hence the name interleukin. There are different kinds of interleukin, so interleukin-1 is the one that is going to cause a fever, and it's also known as an endogenous pyrogen. Now we'll talk a little bit about the adaptive or the specific defenses. That is your third line of defense, and like I said, these are more specific. When you use them, that means that you are now immune to that pathogen. In order for it to function correctly, your immune system has to have that ability to differentiate between what is your cell or your own self cells and identify those and not attack it, while it should have the ability to identify non-self cells. Okay? And it does that through different proteins and carbs that are found on the cell membranes of all of our cells. Those proteins are known as antigens. So an antigen is, again, found on surface membranes, on cell membranes of all of our cells. They could be made out of different proteins or different carbs. For example, the antigens that we talked about when we discussed our different blood groups. So we said we've got antigen A and antigen B and so on. These are examples of antigens. These are self-antigens, though. When an antigen is a non-self molecule, though, if the body identifies these surface molecules as foreign, that is going to produce an immune response. Okay, and that is what is going to trigger our adaptive defenses, whether they are cellular or humoral immune responses. So your cellular responses are performed by your T lymphocytes. The humoral immune response are performed by your B lymphocytes. This kind of shows you all of the lines of defenses. So the pathogen tries to enter your body. Your first line is your mechanical barrier, the skin and the mucous membrane. But what happens if the pathogen actually enters the body? Well, we have the secondary lines of defense, which are your chemical barriers, natural killer cells, inflammation, phagocytosis, and fever. You also have another line of defense, and that's your third line, which are your cellular and humoral immune response. These can actually work at the same time. So you see here that they're kind of on the same level, time-wise, meaning that you don't have to wait for this to fail in order to activate this. They can both be activated at the same time. So we already talked about what an antigen is. An antigen is a receptor that is found on, well, it does say here on the lymphocytes, but it's found on all of our cells. And it helps us identify and recognize our cells, or differentiate between self and non-self antigens. So the antigens can be made out of proteins, polysaccharides, glycoproteins, glycolipids. The bigger the antigen is, the bigger the immune response to it. So the most effective antigens are the ones that are very large and complex. The antigens that are really small, and they cannot induce an immune response on their own, but they would have to actually attach to a bigger protein in order to, or a bigger molecule in the body in order to elicit an immune response. Those are known as haptens. So since the lymphocytes are the cells of the third line of defense, we want to understand where they're made. So lymphocytes, we make lymphocytes throughout our whole life. It starts during fetal development. It all starts in the red bone marrow. So we are going to make these unspecialized lymphocyte precursors, which basically means we don't know if these are going to end up being T or B lymphocytes. It's not until some of them settle in the thymus gland, and those, again, under the effect of thymosin hormone, are going to differentiate into T lymphocytes, while others are going to reach they'll differentiate in the bone marrow and reach the spleen and lymph nodes. Those are going to differentiate into your B lymphocytes. Okay, so your T lymphocytes, again, those specialize in the thymus, and the majority of our lymphocytes, so about 70 to 80% of our lymphocytes in our bloodstream are of the T kind. And these T cells, after they mature in your thymus gland, they are going to go to settle down in the lymph nodes, thoracic duct, white pulp of the spleen. While the B lymphocytes, those mature in your bone marrow. The B actually does not stand for bone marrow, but it does help. B stands for bursa fabricus in chickens. So we first discovered these in chickens, but then we found out that we have the exact same process, and we kept that name B cells. So these B cells, again, make up about a quarter of the lymphocytes in our bloodstream. They are going to end up living in your lymph nodes, spleen, bone marrow, and the intestinal lining as well. You can see here kind of like that cycle. So we'll start off with the precursors in the bone marrow. If it remains in the bone marrow and matures there, we'll end up making B cells, and then these B cells have to be transported by your blood in order to nestle into whether it's the spleen or the lymph nodes or the intestine. While these precursors, if they end up reaching the thymus gland under the control or under the effect of thymosin hormone, these are going to mature into T lymphocytes. And then T lymphocytes can end up also nestling here in your spleens, in the lymph nodes. So how is it that the T cell and B cell, how do they perform this immune response? So we need to understand a couple of things first. Okay, so in order to understand how the immune system works, the interaction between the T and the B lymphocytes, we need to know that these lymphocytes are inactive and they have to be activated in order to respond to an antigen. We'll start off by activating your T lymphocytes, the T helper cell. So let's first introduce the different kinds of T lymphocytes. We have a helper T cell. Okay, the helper T cell is the one that is going to activate everything else, activate all of those other T lymphocytes. It'll also help in the activation of B lymphocytes. We have a cytotoxic T cell, and those are able to attack virally infected cells or cancerous cells. So there is an interaction between cytotoxic T cells and interferon. And then we've got memory T cells, and those produce provide future immune protection. So these are cells that we are going to make and that we could have them forever, meaning that if we get reinfected by that exact same virus 10 or 15 years later, that memory T cell is going to be able to identify it right away and produce a fast immune response. This is really how immunity works, by the production of these memory T cells. That's how our shots and vaccines and so on work. So now knowing the different kinds of T cells, let's try to figure out how do we activate that first and most important cell, which is the helper T cell. So I'm going to kind of skip down to this image right here. We have what are known as antigen presenting cells. Antigen presenting cells are macrophages or B lymphocytes or other cells of the immune system that engulf or eat up the germ. This right here is your germ. This is a huge macrophage. It'll digest the whole germ and display the part that identifies it. So for each organism, it has like kind of a thumbprint to identify it, and the APC, or the antigen presenting cell, is going to again digest the whole germ. Okay, we don't have to use the whole germ. It'll eat it up, break it down into its smaller components, and just display the part of the germ that identifies it. That is the thumbprint of that germ. Now we know that what kind of germ we're trying to deal with. Okay, so that is why it's known as the antigen presenting cell. It'll display the part of the germ that identifies that, and that's known as displaying the antigen. It displays the antigen on this little tiny purple molecule that is known as the MHC, or the major histocompatibility complex. All of our cells have MHCs, whether it is your immune cells or other cells of the body. The function of the MHC here in the immune system is to carry that antigen and expose it to the rest of the immune system. So you'll see here that the MHC carrying that antigen is going to present it to your cytotoxic T cells. Now the cytotoxic T cells is able to identify that there's an antigen, because normally it would be interacting with the MHC, which is your cell, which is a self-antigen. But now that this self-antigen is no longer there and is masked by this non-self-antigen, that'll activate the cytotoxic T cell. Now, and the helper T cell. Okay, so we'll talk first actually about the helper T cell. Now that the helper T cell is able to identify it, it'll start proliferating, making different copies and releasing different chemicals. So we'll make cytokines. Cytokines are going to activate your B lymphocytes. And these helper T cells are also going to make interleukin. Interleukin will activate even further your cytotoxic T cells. Cytotoxic T cells are going to start proliferating and differentiating into different things. So part of them are going to remain as cytotoxic T cells to kill the germ now, while others are going to become memory T cells that will remain in our blood. Again, some of those can remain for 10, 15, 20, even for, can sometimes maybe provide lifelong immunity. Okay, so that is the side on your T cells. What about the side, what is going on with the B lymphocytes? Right here you have your B cells with the antibodies, on the, with the antigenic parts. These are the part of your B cells themselves. This B cell is able to recognize that there is a germ that came in, and you can see here that there's that interaction between the antigen and the antigen receptor. The only problem with B cells that now that they know there's an antigen, they need to be activated. So they are activated by the cytokines that came from those T helper cells. Now that there's an activated B cell, this activated B cell is going to proliferate and make clones of these B cells. Some of those B cells are going to change into plasma cells. These plasma cells are your antibody factories, or antibody secreting cells. Now these antibodies are going to go ahead and attack the antigen, while other parts of the B lymphocytes are going to differentiate into memory cells. These memory cells are dormant and remain in our bloodstream again become part of the immune system for long periods of time, and this is how we become immune if we are attacked by that antigen one more time. Now for the different kinds of cytokines, or the chemicals that can come out, we have the colony stimulating factors, and those stimulate your bone marrow to produce more lymphocytes. We have interferon. Those we kind of associated interferon with the attacking or blocking of viral replication and attacking cancer cells. Interleukins, those have very different responses to them. We talked about interleukin 1 that initiates, and we've called that the endogenous pyrogen that leads to a fever. Other interleukins can control how lymphocytes differentiate and proliferate. There's another cytokine known as TNF, or tumor necrosis factor, and that stops tumor growth. And again, this is another kind of cytokine that is being looked into and researched to use our own immune system to attack cancer cells instead of using chemotherapy. So those interferons and TNF, you might want to kind of, keep an eye on those. Those are they have the potential to be huge in the next upcoming years. So what does an antibody look like? Antibody is Y-shaped. We have these very longer proteins. Those are known as your heavy chains. Those are the purple ones. We have short chains. Those are the blue ones. They are attached by these sulfide bonds. This part that does not attach to the antigen is a constant region, which means that no matter what kind of antibody it is, they all look the same. But this part up here is the part that has to be specific to different antigens. So this would be an antigen binding site. Okay. Remember we called the antibodies, we called those were your gamma globulins. And these antibodies, generally speaking, are specific to antigens. Okay, that is why this is part of the specific immunity. It's not part of your non-specific or your more innate immunity. The different kinds of antibodies, there are 5 different major kinds. There's immunoglobulin G, A, M, E, and D. Okay, in order to remember them, in order they're gamed, okay, G, A, M, E, D. You can kind of switch these two, that's fine. So the majority of the immunoglobulins we have are immunoglobulin G. About 80% of our antibodies are immunoglobulin G. You'll find them in your plasma and in tissue fluid. Immunoglobulin G is one of the immunoglobulins that can activate complement. So complement, again, is kind of your very aggressive kind of immune system. Once you activate the complement system, your, again, immune system is now on steroids, okay? So there are only 2 immunoglobulins that have the ability to activate complement, and that's immunoglobulin G and immunoglobulin M. Okay, so we'll talk a little bit about immunoglobulin A. Those are found in your exocrine gland secretion. So you'll find them in sweat, for instance. Any secretion that comes out of an exocrine gland will have an IgA component. It defends against bacteria and viruses. So your sweat, our sweat has antibodies in it. Okay. Another immunoglobulin that is found in exocrine glands secretions are immunoglobulin E's. Okay, IgEs, those help in inflammation and allergic responses. Immunoglobulin M's or IgM's is found in plasma, and these are the kind of antibodies that are found that are part of the ABO blood system. So when we talk about antibody A or antibody B, in the terms of your, in terms of blood transfusion, this is the kind of antibody that is going to attack those A and B antigens. So those are, the IgMs are responsible for the mismatched blood transfusions. And again, remember that it activates complement. IgD, these are found on the surfaces of B lymphocytes, and those help in B cell activation. So if you kind of go back to this image right here, those are the ones that we're talking about. Those are your IgDs. They're found on the surface of the B lymphocytes. So to kind of sum up the major immunoglobulins, IgG and M are the ones that can activate complement. A and E, or your vowel ones, those are the ones that are found in exocrine secretions. The one that is responsible for the ABO or the mismatched blood transfusion reactions would be the IgMs. So the action of antibodies, it can either attack the antigen directly, or it can activate the complement system, or it can stimulate local inflammatory response to kind of, again, to prevent the spread of the infection. Now there's a lot, immunotherapy is a relatively new area of science, where we want to use our own immune systems to help us to fight diseases and to help us, fight maybe different cancers, as opposed to introducing different chemicals into our body. Okay, so we are maybe trying to activate the humoral immune response by making more antibodies or our T cells to produce more cytokines and provide more protection. We can also mass produce now what are known as monoclonal antibodies, or these mAbs. These are more specific B cells that are made makes a certain type of antibodies. That's why they're known as monoclonal, because we're making just one kind of antibody. So we use those for research. We try to use those to detect new or the recurrence of cancers. They're used in different medications in order to for treatment of autoimmune diseases. For example, Humira, which is adalimumab. The mAb means monoclonal antibody. So any pharmaceutical agent out there that ends with a mAb is made using this technology of monoclonal antibodies. Cytokines, we can mass produce cytokines now due to the technology of recombinant DNA. And we're trying to use these cytokines to treat different cancers like leukemia, maybe treat MS, which is an autoimmune disease, hepatitis, genital warts, melanomas, and kidney cancers. Now for the different kinds of immune responses, we've got a primary and a secondary immune response. So the primary immune response, that is when your body is introduced the very first time to that antigen. So you see here the very first exposure. Okay, your body's response to it kind of peaks at about 2 weeks. Okay, and this is how long it takes your body to defend you against that new antigen. And then that primary response kind of tapers down a little bit. Okay, so you see here that the first antibody starts to appear at about day 5. So you've got 5 days where your body is trying to use its general non-specific defenses to attack that germ. It's not until 5 to 7 days later where it started to make antibodies and so on. So your primary response is a very slow one, okay, and it fades away right away. While your secondary immune response, now that you have developed those memory cells, whether T or B memory cells, as soon as that antigen tries to get, comes back in, your memory cells are able to identify it right away, and you can see that it shoots up the production of antibodies against it, and it protects you within the matter of about, 1 to 2 days. You'll find the antibodies are starting to appear in your bloodstream. So how do we classify immunity on a practical basis? So we have acquired depending on where the source is. So if I obtained the immunity through natural courses, so for example, getting sick, okay, and I recovered from it, so that now that immunity was through nature, or maybe I got the immunity from in the fetus, okay, from the mom through the placenta, or breastfeeding as an infant, okay? So all of these are natural processes. If I obtain the immunity through artificial processes like an injection, that would be considered artificially acquired immunity. Now, passive and active immunity depends on whether the antibodies that were made, whether the immune system made it yourself, so your immune system was actively involved in making those antibodies. That is known as active immunity. This is a more permanent kind of immunity because you have activated your immune cells and you have made memory cells. Passive immunity, that is a temporary kind of immunity where you got a shot that that shot had antibodies in it. So the body was not exposed to antigens whatsoever. Your immune system was not involved in making those antibodies, but your body was given those antibodies very passively in order to protect you now. Okay, so that is a passive kind of immunity. So let's take a couple of examples. So let's say, for instance, that a person got infected. So a person did not maybe get a measles shot as a kid. They got exposed to it. They got the measles, and then it stimulated their own immune response, and now they are now immune to measles. So that is known as naturally acquired active immunity. Okay. You could have the exposure to the vaccine. So a person gets the weakened or the attenuated form of the measles, okay, in the form of an injection, but they do not get sick, yet they are also stimulating their immune system, but without getting the symptoms of the disease. That is known as artificially acquired active immunity. In naturally acquired passive immunity, that is where the antibodies are just passed on from mom to the fetus, okay, or through the breast milk. Colostrum is that very first part of breast milk, the very first day or two of breastfeeding. Those are very rich in antibodies. Okay, that'll provide short-term immunity up until the baby's immune system is up and running. It'll provide passive immunity because those antibodies were pre-made, by mom. Then there's artificially acquired passive immunity where antibodies are injected into that person through an injection. So injection of an antiserum, or that antiserum will either have antibodies or antitoxin. Again, that's a short-term immunity, and it does not stimulate the immune response of the person. Now we've got different hypersensitivity reactions. So we do like our immune system, but sometimes it overreacts, and that overreaction is known as hypersensitivity, which is an exaggerated immune response to a non-harmful antigen. And there are 4 different kinds depending on how your immune system reacts. So we are going to discuss the 4 different kinds of hypersensitivity reactions. The first type is known as type 1, or immediate reaction. And those are just your regular allergies that occurs in minutes of contact with that allergen. Okay, it's mediated by these immunoglobulin E's. Okay, for example, the production of hives, hay fever, asthma, eczema, gastric disturbances, or even anaphylactic shock, which is a life-threatening severe form of type 1 hypersensitivity. So these are, again, immediate reaction that occurs within seconds or minutes of exposure to what you're allergic to. Compare that to type 2 hypersensitivity. That is dependent on the presence of the antigen and the antibody together. So for example, in the transfusion reaction where a mismatched transfusion, we'll have that interaction between the antigen and the antibody, and that can lead to what is known as type 2 hypersensitivity. It'll lead to phagocytosis and the breakdown of those different cells. Type 3 hypersensitivity also depends on the interaction between an antigen and antibody. It makes that immune complex, which is antigen plus antibody together is now an immune complex, but it doesn't destroy the cells right away. It'll deposit on certain tissues like joints, for example, and destroy those joints. Okay, so it'll damage the tissue by phagocytosis, activation of complement. And unfortunately, the body does not have the ability to clear or get rid of those antibody complexes. So for example, RA, or rheumatoid arthritis, is an example of those type 3 hypersensitivity. Type 3 hypersensitivity is more of the autoimmune kind of reaction. Type 4, or delayed reaction, that can happen to anybody. We are exposed due to the overexposure to, or repeated exposure of your skin to an allergen. For example, if you got a tuberculin test done before, okay, the TB test that is done where they inject a little bit of the, attenuated or even dead TB underneath the skin, intradermal into the skin, and then they ask you to come 2 days later to see if there's a reaction or not. So you come in 2 days later, 48 hours later, and that's why it's known as a delayed reaction. And this kind of allergy is due to T lymphocytes. Transplantation and tissue rejection also ties into those mismatching of different antigens. Okay, so we can transplant different tissue and different organs. Here are kind of, some examples. But if the donor's tissue, if those are recognized as foreign, this is when a tissue rejection reaction is going to occur. So the recipient's immune system will start destroying that foreign tissue. That's why you really want to match as much as possible the MHC antigens of those cells between the donor and the recipient. So the more they match, okay, the lower the risk of a rejection. We sometimes give immunosuppressive drugs in order to prevent rejection from occurring. These are different kind of transplant types. So there's an isograft, which is transplant between identical twins. Okay, autograft is where you get a graft from your own self. So for example, an area that has been exposed to burn, you can take a skin graft from one part of the body and put it into another, and to replace that burned skin. An allograft would be grafts between the same species. So human, kidney transplant from a relative or a closely matched donor. Okay, so in between humans. A xenograft would be transplant from a different species, for example, heart valves that we get from pigs. Autografts and isografts, so these two, whether you're getting the tissue from yourself or from an identical twin, those are not going to cause rejection reactions. These two would or have the potential to cause a rejection reaction. Autoimmunity, again, all depends on those the ability of the immune system to recognize self from non-self antigens. And the body, if the body makes antibodies against its own antigens, those are known as autoantibodies. We have lots and lots of immune diseases or autoimmune diseases that, we'll show on the next slide. There are different theories behind why our immune system is no longer able to identify our own antigens, but nothing has really been established so far. So these are a list. This is a list of the different autoimmune diseases. Okay, those are just examples. So glomerulonephritis would be in the kidney. Graves' disease would be against the thyroid tissue. Type 1 diabetes, okay, against your pancreatic beta cells. Hemolytic anemia against your RBCs. Multiple sclerosis or MS those are autoantibodies against the myelin sheath. MG or myasthenia gravis, those are antibodies against the receptors on the skeletal muscle fibers. Pernicious anemia are autoantibodies against the certain cells inside of the stomach, and we'll talk about that more when we get to the digestive system. Rheumatic fever are autoantibodies against heart valves. Rheumatoid arthritis, we talked about that. That would be autoantibodies against your joints. SLE or systemic lupus erythematosus, those autoantibodies against your own connective tissue, and that's why you'll find that there is a widespread of symptoms because it's connective tissue throughout the whole body. Ulcerative colitis would be against colon cells. Lifespan changes that happen to the immune system, we kind of already talked about how the thymus gland tends to shrink, and that is why we have a higher risk of infection and cancers as we age. T cell numbers tend to decrease, B cell numbers not as much, but the activity, so the ability to perform their actions does decline. The antibody response to antigen becomes slower. As we age, IgA and IgGs increase, but the IgM and IgEs decrease. Okay, sometimes elderly, we might not be able to give them things that further suppresses their immune system, like chemotherapy, in order to treat their cancers due to the fact that their immunity is already suppressed. Now, a little bit about immunity breakdown, HIV positive or AIDS patients. So HIV, or a human immunodeficiency virus, is the virus that has the potential to break down the immune system. Okay, it could be silent for many years, so somebody might be HIV positive for years on without any symptoms, but when it does start attacking the immune system and leading to signs and symptoms, that's when we are going to call it AIDS. The HIV attacks mainly your helper T cells. Okay, so remember that the helper T cells were the cells that were responsible for activation of the rest of your cells. So activation of B lymphocytes, activation of cytotoxic. So when when HIV, kind of paralyzes your helper T cells, then you have basically paralyzed the whole immune system. Okay, The causes of death would be due to the loss of the immune response against any pathogen, and cancer start to develop in these patients. Mode of transmission of HIV, sexual contact, contaminated needles, birth or milk from infected moms, okay, or received infected blood or tissues from donor. That concludes chapter 16.